There's no question that we'll be able to use this technology to eliminate diabetes altogether within our lifetime, if not within the next 10, 15 years.
Dr. Robert Lanza is the Vice President of Medical and Scientific Development at Advanced Cell Technology, a biotech company in Worcester, Massachusetts. Dr. Lanza is leading a team of some of the world's leading biologists to explore the possibilities cloning holds. Using several proprietary techniques, Lanza's team has successfully cloned several large farm animals and is exploring possibilities that might have been considered more science fiction than science just a few short years ago.
We'll say, for instance, that you had heart disease. What we would use is this technology to basically generate new heart cells so that we could transplant those back into the body, and they would of course be your own cells and they wouldn't reject. And the way we actually do that is we would take a skin cell, say a cheek swab or a little piece of your skin, and we would grow those cells up in the Petri dish. And then we would take one of those cells and we would put it next to an empty egg cell that has had all of the DNA removed. And then when they're side by side, you send a little electrical fuse, a little current through the two cells, and what that does is it damages the membrane between the two cells so that the two cells fuse, and that the DNA from the cell you want to clone is dumped into the empty egg, and then you fool that egg into thinking it's been fertilized, and it starts to divide.
On July 5, 1996, a sheep was born in Scotland that would begin a revolution. For the first time in history, a mammal had been cloned from a single cell taken from an adult animal. A worldwide controversy was spawned, but at the same time, a world of remarkable possibilities came to light. 4 years later, scientists, ethicists, and theologians gathered in Oak Ridge, Tennessee, to consider the future of medical technology, the science of cloning.
Just this past few weeks has seen, as Dr. Lanza described yesterday, an incredible advance in technologies associated with cellular and whole animal cloning in the world of mammal biology.
When Dolly appeared on the world stage, there was no shortage of controversy. Much of the controversy has since been focused on the possibility that soon a human being might be cloned. Few reputable scientists consider it ethical to do this. Most research is focused on the use of cloning technology to cure humanity's most dreaded diseases. And while the promise is great, there is still controversy.
Within a few days, the New York Times, for example, had sent, not just the one reporter, but they sent somebody from Moscow, somebody from Frankfurt, somebody from New York, and a photographer. 4 heavy-hitting journalists, all from the one newspaper. That sort of scale of activity we just didn't anticipate.
There are people who are concerned that the origin of these ES cells, came from what we call a pre-implantation embryo that had the potential, should it be implanted into the uterus, to become a person. By the same token, virtually any cell of the body has the genetic potential to become a human being. So I think that it really raises the question as to, you have these cells coming from these embryos from IVF clinics that would have been discarded in any case. So you have cells that are growing in an incubator that will continue growing indefinitely, and you have 2 choices. You can throw them away, or you can use them to create cells that would hopefully one day be able to cure, for instance, diabetes or treat various other diseases. And one thing I should point out is already most of the vaccines that you and I have received, be it rabies or be it polio or any of the routine vaccines that you use in childhood, those vaccines are derived from a fetal cell that was derived from an aborted fetus. It's just a cell that continues to grow and grow and grow, and these vaccines are derived from those cells. So the question is you have these millions and millions of cells that continue to grow. Do you throw them away, or do you use them to save human lives?
When Dolly was cloned, her creator Ian Wilmot was quoted as saying, "Human cloning has grabbed people's imagination, but that is merely a diversion, and one we personally regret and find distasteful. Indeed, to become concerned only with who will be the first to clone a human being and when it will happen is to entirely miss out on the possibilities. consider the following uses of cloning technology when it is combined with associated technologies of genomics and genetic engineering. New genes can be spliced into embryonic cells to produce animals with desired characteristics. Animals might be produced that contain human genes, creating human proteins such as clotting factor for hemophiliacs. Identical animals might be created to increase accuracy of tests of new drugs. Most of the excitement about cloning surrounds the creation and use of stem cells to cure disease. Stem cells can be derived from different types of tissue in the body, but naturally occurring stem cells are limited in their ability to heal damaged organs. The holy grail of stem cell research is to create a culture of totipotent stem cells. Totipotency is a term used to indicate a stem cell's ability to become any type of cell in the human body. In nature, when a sperm cell fertilizes an egg cell, the resulting cell is totipotent. It can and will divide again and again to become all of the highly specialized and differentiated cells that comprise the body. Normal adult cells no longer have the ability to become any other type of cell. Adult stem cells can become certain kinds, but not all kinds. So where does one go to find totipotent stem cells? They can be gathered from frozen human embryos discarded from in vitro fertilization clinics, or they can be cloned through a specialized process in the laboratory. Advanced Cell Technology has patented a process by which adult cells can be made into totipotent stem cells.
Well, what we're talking about in this particular instance is taking a body cell from an adult, a completely differentiated cell, and bringing it back in time through the molecules in the egg so that you've now got what we call a totipotent cell. It's basically a master cell that has the ability to become virtually every type of cell in the body.
Because before the birth of Dolly, people had understood, had believed that the genetic information in adult cells was fixed. That the process of going from the single cell of an egg through to all of the different tissues of an adult meant so many changes in the genetic information, such complex changes and so permanently fixed changes, that it was simply not possible to go back to the beginning. And what we showed is that you can.
There are two types of stem cells. There are the embryonic stem cells that are coming from what we call a blastocyst, a pre-implantation embryo, so to speak. And then there are cells that are coming from the adult. And these adult stem cells, say it's from the fat or from hematopoietic stem cells, do have the ability to change into different cell types. The big question there is whether or not you can actually get enough of these cells to be of any value. But even more importantly is whether you can get the kind of cell that would have some therapeutic value at all. We do know that you can get hematopoietic stem cells that would be of value for reconstituting, for instance, some of the immune system. But we would be very doubtful whether or not you could get an adult stem cell, for instance, that would create insulin-producing cells that would be of any value in diabetes.
It's a surprise to people initially that the same technique that you can use to make copies can also be used to make precise genetic changes, because you make the changes in the cells before you do nuclear transfer. And it was this application which was actually our original motivation to start this research, to be able to make changes in animals, to produce proteins needed to treat human disease, to produce organs from pigs to transfer into people, and in order to understand the function of particular genes. This is what motivated us in the first place.
Again, there are millions of people who are dying, and we do know for a fact that the embryonic stem cells have the potential not only to make insulin-producing cells, but virtually every tissue and cell of the body. So that the master cell, those are the cells that we know for sure have the greatest potential to differentiate into the cell types that we want. We have stem cells that will, for instance, in one dish, you'll have a whole dish of beating heart cells, and then we'll have another dish full of neurons that are making dopamine, the very cells that you would need to cure patients with Parkinson's disease. In another dish, we have neurons that are motor neurons, which could be used for people with spinal cord injuries. Other kinds of neurons, for instance, could be used in that instance as well. So we do already know that we can turn these cells into liver cells, into heart cells. And not only can we make those cells, but then using a technique known as tissue engineering, we can reconstitute those cells into more complex tissues and structures, say into an entire kidney, or even eventually in a number of years into an entire heart. So we can grow the cells in the lab, and then by using tissue engineering, reconstitute them into bones and blood vessels. And already this has been done. We already can grow an entire organ. Tony Atala at Children's Hospital, for instance, has grown an entire bladder. He's put them back into dogs, and they've worked very nicely. So we do know using this technology in conjunction with tissue engineering is going to be the way of the future. So when you and I grow old and start to develop these various diseases, that's how our organs are going to be replaced. You're going to get a cheek swab or a little piece of skin, and we will grow you up a new kidney.